Hello everyone, welcome to Pharma Icon, and the today's topic is about the antiviral drugs and their mode of action. So, first of all, what are antiviral drugs? So, the antiviral drugs, these are mostly used for the treatment and prevention of the viral infections. Okay, they are opposite the action of the viral conditions. Okay, so these are mostly DNA polymerase inhibitors. Okay, these are mostly DNA polymerase inhibitors. Okay, these are mostly DNA polymerase inhibitors. So first of all, the antiviral drugs is acyclovir. Here the main important thing is the antiviral drugs are they end with the suffix vir. Vir stands for viral. Okay, the antiviral drugs are ends with the suffix vir. The first drug is acyclovir. The acyclovir is mostly inhibits the treatment and prevention of diseases like herpes simplex, varicella zoster and herpes zoster. These are mostly inhibits the DNA synthesis in the that virus molecule. Okay, these acyclovir is guanosine derivatives. What is guanosine? Guanosine is a molecule which is the structurally similar to the guanine. Okay, guanosine is structurally similar to the structure of guanine, and these guanosine also helps in the treatment and prevention of the cancer. Okay, how this guanosine helps in the cancer treatments? Because the guanosine itself it act as a DNA inhibiting agent na? So it is break down the DNA synthesis. So what is a cancer? In cancer the irregular growth of the cells means continuously the cells are growing. So these guanosine when we used in the cancer treatment, it is reduces the DNA, it is reduces the cells growth and damage the DNA. So hence it leads to the breakdown of the growth of the cells. Next, valcyclovir. Valcyclovir it can also act as a DNA synthesis inhibitor. Okay, here what are DNA polymerase inhibitors? The DNA polymerase inhibitors are mostly the DNA polymerase, it is an enzyme, the enzyme which is mostly inhibit the activity of the DNA synthesis. Okay, the polymerase is a type of an enzyme which is mostly inhibit the DNA synthesis. Okay, so it is disrupts the DNA replication. So the DNA replication can be disturbed, it is automatically leads to the formation of the diseases. Okay, next, then we go to the first, what is a nucleoside. Okay, so this is about the different types of DNA polymerase inhibitor drugs. They are ganciclovir, vanciclovir, penciclovir, famciclovir, edoxyridine, trifluridine, ribavirin, foscamate and vidaribin. Okay, these are the different types of DNA polymerase inhibitors. And the next one is nucleoside reverse transcriptase inhibitors. Okay, what is nucleoside? The nucleoside is a molecule which contains 5 carbon along with 1 sugar and a nucleoside base. Okay, nucleoside base, and which is mostly for the DNA and RNA. These nucleoside is important for the DNA and RNA. And then what is reverse transcriptase? So the reverse transcriptase, which means they blocks the enzyme and reverse transcriptase. Okay, reverse means actually first the DNA is converted to RNA, but in reverse transcriptase means the RNA is converts into the DNA. Okay, that is the action is in reverse. Okay, next, what are the drugs act as a nucleotide reverse transcriptase inhibitors are abacavir, zidovudine, dianosine, zalcitabine, stavudine and lamivudine. So these are the different types of the drugs. These are act as a nucleoside reverse transcriptase enzyme. Okay, these are mostly used for the treatment of HIV infections. Okay, these are mostly used for HIV infection. Okay, and the next one, deoxy. So these are the guanosine analogues, thymidine analogues, and deoxydenosine analogues. These are a type of purine nucleoside. Okay, purine nucleosides, what they are done? These purine nucleosides, these are inhibits the DNA synthesis. Okay, DNA synthesis, they process to be inhibited by the deoxydenosine. Okay, next, what are thymidine analogues? The thymidine analogues are one of the synthetic compounds. They are mostly used to replace the thymidine. Okay, they are replace the thymidine at the cell cycle. Okay, that cell cycle is at which phase? At S phase. They are mostly replace the thymidine. And next one, what is cytosine? The cytosine is a pyrimidine compound base which is present in the DNA and RNA, that is A with T and G with C. Adenosine with the thymine and cytosine with the guanine. Okay, and the next one, and they carry the genetic information in cells. Okay, next, what are nucleotide reverse transcriptase inhibitors? So the nucleotide reverse transcriptase inhibitors, the nucleotide which is mostly consisting of a nitrogen ring, a base and a phosphate group along with the sugar molecule. Okay, there will be a small difference from the nucleoside and the nucleotide. Okay, sugar molecule will be present. Next, example for nucleotide reverse transcriptase inhibitors are tenflovir. This is one of the important drug, act as a nucleotide reverse transcriptase inhibitor. And the next are non-nucleoside reverse transcriptase inhibitors. So the non-nucleoside reverse transcriptase inhibitors, the example for these drugs are ephiramides, and that is short form EFZ, and nevirapine and detavirtin. So these are the three important drugs. These act as a NNRTI, means non-nucleoside reverse transcriptase inhibitors. And the next one are protease inhibitors. So what are called protease inhibitors? The protease means a type of enzymes. These are mostly prevents the breakdown of proteins. Protease, pro means proteins breakdown. Okay, these are mostly used for the treatment and prevention of the viral diseases. Okay, what are viral diseases like HIV, AIDS? Okay, they are mostly reduce the amount of HIV in the blood. Okay, these drugs, the protease inhibitors, are mostly reduce the amount of blood in the HIV. Okay, next, the drugs are the saquinavir, indinavir, ritonavir, ampinavir. Okay, these are the different types of drugs act as a protease inhibitors. Okay, that's all for this session. Thank you, and please do subscribe Pharma Icon.